a lecture for metabolism, we're going to take a look at the ETS. This is the image that can show up on your exam. So this one's a little harder for me to clean up, but I do. Here's what you're responsible for knowing. On this, you would need to know the names of these three complexes. So they're here in these purpley kind of lavendery boxes. The NADH dehydrogenase complex, the cytochrome B-C prime complex, and the cytochrome oxidase complex. They refer to NADH, cytochrome B-C, and the cytochrome oxidase complex. That's these three carriers here. Then you have ATP synthase that is right here. So you do need to know that's the enzyme and the function of this structure right here. You need to know that this is the inner mitochondrial membrane. You don't really label that, but recognize what you're looking at. You need to understand where the matrix is, which is the lighter colored peach color here, and the darker kind of rosier color is the space between the inner and outer membranes. So understand we're working right along the membrane of the mitochondria. If I back it up and look at the picture of our mitochondria, we're on this gold line right here that we're moving along. This process of complexes and enzyme actions are repeated numerous times all along that cristae, or that inner membrane of the mitochondria. Okay, so you've got proton pumps is what these complexes are. You've got all these carrier molecules, and so the thing that you have to identify, the names of these things down here. Then you see here where the NADH is going to join in, and electrons are going to move along here, and they're they're going to be carried all along where these pink arrows are. So the electrons that come along that are released from this remember, hydrogen's an electron and a proton. So the electrons get captured and managed within here, and we actually neutralize those electrons. Electrons are really free radicals. They can be very damaging. And so a proper body functions by having all of them controlled right here, and then they're neutralized in the formation of water. So we sometimes refer to metabolic water that's produced in the body. If you never drink any water, you still urinate at some point during the day. You brought in no fluids, you'd say, how can the bladder still be producing fluids or excess water? And that comes from metabolic water of the metabolism. So you're still producing a net water gain from as long as you're still metabolically functioning. And so, which basically means you're alive. So this water is a neutralization. So the electrons are along these pink lines. So you'd have to just draw, I'd ask you to draw where the electrons, indicate with some arrows where the electrons go. And they're all right along here till water's formed. And then I'd ask you to draw, well, where do the hydrogens go? Well, the hydrogens get pumped up here. And you'll see that NADH, that molecule, that electron carrier, enters the process right here by the number 1. And that by this proton pump, and pumps across the hydrogens with the electrons being captured and carried by these various carriers. I do not ask you to label all of these little carriers. So the FMN, the Q, the cytochrome B, C prime, or C1, the cytochrome C, A, and A3. I don't ask you to label those. So usually they're not labeled at all, but I don't ask you to know the specific ones. I want you to know the complexes, synthase, where NADH, I actually ask you, draw NADH function. See how it's being, this is an oxidation reduction reaction right here, because now NADH is oxidized to NAD. It can now be reused. And the hydrogens are pumped up here into this space, and the electrons are housed within the membrane. Okay? Notice the blue here, FADH2 enters in here. It misses the first proton pump. You'll see that FADH2 actually produces slightly fewer ATP than NADH. Okay, and that's why they chose 3 ATP here for a single NADH, but FADH2 only produces 2 ATP, and we'll look at that more closely in a minute. What I want to do before I even go any further with how this is really all functioning is let you listen to this video, and I'm really hoping it plays in our little recording, or on this lecture I'm recording here. So I'm escaping out, And this is the link that was right there on the PowerPoint page. So we're going to play this. It's not very long. If it doesn't play well in the lecture, I'm not going to re-record this lecture. You can still see what's happening even if you can't hear it. You can read what's along the bottom, and you yourself can go to this link and play this link.
In the mitochondria, the energy stored in NADH is used to generate a proton gradient across the mitochondrial membrane, and the energy of the proton gradient is used to make ATP. When glucose is oxidized during glycolysis and the Krebs cycle, the coenzymes NAD and FAD are reduced to NADH and FADH2. Inside the mitochondrial matrix, the electrons from NADH are transferred to the electron carrier coenzyme Q by NADH dehydrogenase, and the protons are transferred across the membrane to the intermembrane space. Coenzyme Q carries the electrons to the cytochrome BC1 complex. As the electrons move from the BC1 to cytochrome C, more protons are carried from the inside to the outside of the membrane. Electrons are also transferred from FADH2 to coenzyme Q, with the protons being transferred across the membrane. Cytochrome C transfers electrons to the cytochrome C oxidase complex. Protons are also transferred to the outside of the membrane by the cytochrome C oxidase complex. The cytochrome oxidase complex then transfers electrons from cytochrome C to oxygen. The terminal electron acceptor and water is formed as the product. The transfer of protons to the intermembrane space generates a proton motive force across the inner membrane of the mitochondria. Since membranes are impermeable to ions, the protons that re-enter the matrix pass through special proton channel proteins called ATP synthase. The energy derived from the movement of these protons is used to synthesize ATP from ADP and phosphate. Formation of ATP by this mechanism is referred to as oxidative phosphorylation.
I'm hoping you could hear that, but again, if not, all of what was narrated is also said along the bottom of the screen, and you can go to this link yourself and view this as well. So I encourage you to do that if you were unable to hear that. So the important factor, though, is understanding here is what you get this process called the proton motive force. Now, these subsequent slides show you other images. This is the one that I'll use on the exam. But there are other images that kind of give you the same kind of idea. I like the mechanism of how it looks like it's kind of spirals through and energy is generated from this, and some hydrogens move from here to here to produce the water. So it talks a little bit about what each of the pumps are. I'm not going to even read all of this through to you. Don't get too caught up in the details here. Things that you would want to know, for instance, is FMN, this that I said you did not need to necessarily label this here, that you didn't have to read. I wouldn't going to put this picture on the exam and ask you to label it, but you should know, for instance, that it comes from riboflavin. So someone whose diet is deficient in their B vitamins is going to have a problem forming this FMN right here. Sometimes we don't think about the importance of iron anywhere but in blood, but iron and sulfur, which sulfur comes from our proteins, this is really important in the diet in order to make sure that you are forming the different pumps that are available here. So iron, sulfur, the BC complex, they require iron and sulfur. In this one here, the third pump, again, we're looking at not having to worry about the specific name of what it is, but copper is important. You think, oh my goodness, how often do I worry about my copper intake? Yet that is a component within the human body. These are elements, these are minerals that are necessary for us in order to have normal function of the ETS. So not something you don't normally see someone with a copper deficiency, but you do see a B complex deficiency, or someone who doesn't eat enough proteins, that doesn't get the sulfurs and other minerals that are necessary for the body to function. So don't get too caught up in this. This is a nice way to walk through how each of these pumps works. The little animation really has the content I want you to understand, and the key here is this thing called that proton motive force. The fact that the hydrogens accumulate outside in that inner outer membrane space. It's kind of like water behind a dam. And so all of those hydrogens, and I like this picture best, all those hydrogens start accumulating up here, and that generates all this energy. Those positive charges want to move back down here so that they can find equilibrium again. It's like water behind a dam. Now we poke a hole in it, and there's energy involved in those hydrogen ions moving through, and now they can be used to generate the ATP. So that's that process. Again, this just kind of walks you through a little more detail on the various pumps. I don't test you real heavily on this at all in terms of all of this, what's the first pump, the second pump. But what I do expect you to understand is that there are nutrients and diet factors involved in this. And so there are also genetic mutations that occur with the formation of these pumps and with these coenzyme carriers. And if they don't exist, or if there are mutations in them, they either were never made or don't exist, metabolism is dramatically affected. They cannot produce ATP. If they cannot produce ATP, go back to the very first lecture and all the things that we needed to release ATP energy for to do. Everything that needs ATP is affected. Every muscle contraction, your sodium potassium pumps, every nerve reaction, everything requires use of energy, and we don't have it if we have a problem here. So that's really the big picture on this. So let's take a look at the summary of all of the actions of everything that's happening here within the mitochondria and even outside. But let's first look inside the mitochondria. We had a total of 10 NADH. Remember, where did they all come from? Backing way up, I'm sliding back fast. You had, in glycolysis, you had 2, right? Then you had 2 here, that's 4. And then from the Krebs cycle, you got 6 more. So you had 10 NADHs and you had 2 FADH2s. So when I take a look at what all those did inside the mitochondria, the 10 NADHs, each one produced 3. So of the 10 NADH, that would be basically, there were 10 total, that's 30. 10 NADH produced 3 ATP per molecule. That's 30 ATP. We'll look at the asterisk in a second. And then the 2 FADH, each FADH produces 2. There are 2 total, so that's 4 ATP. Right here, I've got 34 ATP. Remember, we produced a net of 2 ATP in glycolysis. That takes us to 36. And then we had 2 ATP in the Krebs, or citric acid. That's 38. So here's 38 ATP. Okay? Now, you may recall that in learning how many ATP are produced from a single glucose molecule, you always got this 36 to 38. There was a variation that's in there. I want you to now know why there's a variation. And that's because the 2 NADHs from glycolysis, they actually cannot get inside the mitochondria. They are too big. So when I slid way back and said, hey, we got 2 from glycolysis, we did. There's energy there. But those molecules cannot get across the mitochondrial membrane. So what do? They donate their electrons, their hydrogen molecules, to a transfer molecule. And these transfer molecules are malate and glycerol. So the malate, we call it the malate shuttle, is most predominant in the liver, kidneys, and heart tissue. So these are real vital organs in our body. And what they do is malate picks up the energy from the NADH, the hydrogens, carries it into the mitochondria, and there is no use of ATP involved at all. It is a molecule that very easily crosses the membrane, carries that energy with it, and we still get 3 ATP. So in this part of the body, liver, kidneys, and heart, we still can get 38 ATP from a single glucose. But glycerol shuttle, which functions in skeletal muscle, neurons, and most other tissues of the body that utilize ATP, and in order to get the glycerol in the mitochondria in those areas of the body, in those tissues, we have to spend a little. So instead of getting 3 ATP per NADH, we have to spend 1 for each molecule, and we only get 2. So we have a net reduction in the total, and that gives us to 36 instead of 38. Okay, so in the glycerol, every NADH that it carries costs them an ATP. So instead of getting 3 from the NADH, we get 2. So that's where we get that 36-38 variation. Important to know, always, can I say it again? Always ask about this on the exam. So please be familiar with this. All right, so here's your summary table that has everything that we got out of all of this productions. These are a reminder of the animations that I've showed you. So if you haven't taken a look at them, I think it's a good idea to take a look at what these are. They're linked also on the Moodle page. And that takes us through what you need to know for metabolism.